I will now introduce Paul Olivier. He is a research associate at the University of Toronto in John Atkinson's lab.
Hello. Do you see my screen?
We do. We see it, and we hear you as well.
Perfect. Okay, great. There's a bit of delay there. Okay. But thank you very much. Just to clarify, I'm actually in Seattle in the Center for Infectious Disease Research, so my bio is a little bit out of date, I guess. So thank you very much. So in our lab today, we're trying to develop a new framework to detect and link protein complexes to specific chromatin interactions. And this is in collaboration with Sanjeev Kumar in the lab and other groups that are part of our grant. And now I'm trying to advance my slides. There we go. So what we're trying to do here is use a base framework that was already put together by the NCDIR pipeline and and modify it so that we can isolate native protein complexes concurrently with the chromatin interactome and to define the sequences that are associated with our proteins that we're pulling down. So the base of this, I just realized I skipped a slide here, so I'm going to go back. So basically, currently there are no techniques that have been developed to provide a coupled information that includes chromatin-chromatin interactions and associated protein complexes that are mediating these interactions. And as you know, chromatin is naturally defined by its bound proteins and their complexes, which orchestrate this three-dimensional folding of the genome, and they initiate functional chromatin interactions such as enhancer-promoter interactions and the formation of chromatin domains. So our overall goal is to develop this new framework, which I gave you a sneak peek on the slide before, to detect and link these protein complexes to specific chromatin interactions. And so we propose an integrated analysis for the simultaneous detection of protein complexes and the genome-wide chromatin interactions mediated by these complexes. So the way we plan on doing this is using this pipeline that was previously developed by the NCDIR, which is taking cells, cells that have been frozen. I don't know if you can see my marker, but they've been flash frozen in liquid nitrogen in order to rapidly preserve their native structure and environment. We then use a technique that was developed to cryomill these cells to break them up to form a grindate, what we call a grindate. And we do this by a motorized grinder until the cells are broken into micron-sized fragments. And I'll have a slide depicting this in a little bit later. We then plan on using glutaraldehyde as a fixation to stabilize these complexes bound to their DNA complexes. Then we would use these antibodies to pull down these complexes, or what in our case here, we prefer to use nanobodies, given that they're high affinity and they're custom and we can make lots of them. We can also mutate them to make them no longer binders, so we could use those as our negative controls. We use these nanobodies to pull down these complexes with magnetic beads, which provide a much lower background of nonspecific binding to the beads. We would then plan on developing a way to then, in our lab, we have to develop it anyways or try it, to ligate, fill and ligate the biotin linkers onto these bound complexes. Then what we have to do is we have to then release these complexes off the beads and then subsequently pull out using avidin beads to pull down the complexes that have DNA and protein on them. So initially we have a mixture of protein bound to DNA and protein not bound to DNA. So we want to enrich for complexes that contain DNA. We then would do mass spectrometric analysis of the protein complexes, and then in parallel we would do paired end sequencing to identify the sequences that are found in these complexes. So the current strategy and overview of what we've done so far and what we're working on right now is that we're working on nanobody generation when we've picked the target of CTCF. we're working on grindate optimization so that we can maximize breaking the cells open and also breaking the DNA into smaller, more manageable size pieces. And then we've also established an interim system while we're developing the nanobodies to optimize this GCCP pipeline protocol. And I'm going to talk a little bit about that now. So the nanobody generation, which we don't have time to talk about that today. As I mentioned, we're focusing on CTCF. We've injected 3 llamas, so we're currently waiting for the purified antibodies, which will be probably in about 9 months' time. But what I want to stress here is that these nanobodies that we generate, we can generate as many high-affinity single-chain nanobodies that we wish, and that we can make these in E. coli and unlimited supplies. And they're small enough to be able to integrate into small complexes and pull down complexes. We're also, I mentioned earlier, we can mutate them so that we can mutate the nanobody so that it no longer binds to the antigen, and we could use that as a negative control. The cryomilling here, we use this custom-designed cryomilling apparatus where we put frozen cells on the top left into this steel chamber here that's been previously cooled with liquid nitrogen that contains steel balls. And we put this into this machine here, which is a shaker, and it'll mill in multiple cycles for long periods of time. And up here in the middle, in the center, you can see this metal jar here with this white powder in there, and that was previously contained these BBs of frozen cells that have been undergone extensive grinding process forming this powder. And then below here is the microscopic image of the grindate, and basically it's hard to see here because we don't have magnification here, but everything has been broken down. There's no nuclei here, and these larger particles that you see here are just aggregates of this grindate that's been put on the slide. And on the right here, you can see that we're getting the unground material versus grinding 1 and grinding 2. So grinding 1 and grinding 2, the difference is the number of hours that we've ground this material. You can start to see significant breakdown of the genomic DNA here on the right, grinding 2. So the interim system for this protocol that we want to optimize, we tag CTCF on the C-terminus with GFP, and we express it under a DOX-inducible system in 293 cells. And on the right panel, you can see that after 24 hours induction, on the top, we can see the GFP CTCF. Levels are still higher than the endogenous, but it's titratable based on the amount of time that we incubate with DOX. In the meantime, we're also going to tag the endogenous CTCF with GFP by using Cas9, and then and also in our interim system, we're going to use, because we're tagging everything with GFP, we're going to use GFP nanobodies for our pull down. And so on this next slide here, we use this grindate, cellular grindate. We can add various lysis buffers to optimize the extraction of the protein, CTCF protein in complex with DNA. We add antibody that's been conjugated to magnetic beads, and then we do the affinity capture of these protein complexes. And we can use a robot here to facilitate the rapid IPs. So these IPs are only happening for 15 minutes, and then the machine runs here to wash our immunoprecipitations, our captures, very fast and reliably and reproducibly over a 10-minute period here. And as you can see here, we've got 8 replicas going here. We can scale this up to 96 well plates as well, and we could do multiple IPs under various conditions and various wash conditions. We then take these IPs, and then we can send them to for mass spec analysis, or we could then elute them in TE or do the proximal ligation steps afterwards. What we've done right now is that we've done some lysis optimization to maximize CTCF complexes and the associated chromatin. So we have 3 different lysis buffers here on the bottom, 1, 2, and 3. And if you look in the next lane, the lane in yellow is our negative control, so that's a nanobody that doesn't bind CTCF. And in red are various nanobodies to GFP. So we have 2 different nanobodies, 16S2 and 94.5. that under these various lysis conditions, you can see CTCF coming down here. It's this bright band just below 160 kilodaltons, and then all the other bands here that are labeled that are stained with Cipro ruby, you can see that there's an enrichment for CTCF and some associated proteins here. We then went and did a rapid capture using lysis buffer condition number 1, and what we did here is we used grindate from minus DOX or DOX treated cells, and you can see that there's an enrichment for proteins coming down in the DOX treated cells with these nanobodies. And on the left panel is 1 nanobody to GFP, on the middle panel is another nanobody to GFP 94.5, and on the right is the negative control. When we do mass spec analysis here, we identified many CTCF interactions. We had about 500 nuclear-specific proteins. And if you actually go in the databases, BioGrid has published that there's 47 CTCF interactors, of which 20 overlap with our pull-down experiment. And this is all preliminary. And these are the proteins here, that the 20 that overlap between these two. We also then wanted to quickly establish whether or not we pull down DNA with these pull downs. And so what I found a paper by Paul Lederman's lab where they were interested in the contribution of human subtelomeric DNA and chromatin organization to the telomere integrity and chromosome end protection. And so they identified CTCF and cohesin complexes and their contribution to this protection. And they used 3 primer sets here, 1, 2, and 3 on the top to detect CTCF binding on the telomeric region of 10Q and XQ. And they found that in the CTCF immunoprecipitations that they enriched for CTCF using primer set number 2 in both cases here. And that's shown in the middle panel graph of each of the 2 figures that I'm showing here from their paper. And if we did the same qPCR, so a rapid CTCF capture followed by qPCR, and it's the same experiment that I showed you before from the same IP that we did the mass spec analysis. So this is now done at the same time, same sample. We see an enrichment for a CTCF. So we think that we've established a quick and a first run at this to pull down simultaneously a DNA complex and then also done mass spec analysis with this. So now what we plan on doing, so to summarize, we have CTCF nanobodies, which are in production. The liquid cryomilling has produced chromatin fragments between 1 to 10 kb in size. We've established an interim system to help detect, to develop and optimize this framework to detect and link these protein complexes to these specific chromatin interactions. And so our first run, we pulled 20 different proteins out here, and we were able to pull down CTCF DNA binding sites. So our future plans are to repeat these affinity captures utilizing cross-linking agents such as glutaraldehyde or formaldehyde. We want to broaden the panel of qPCR primer sets to get a better understanding of CTCF, or maybe even do a deep sequence. We want to set up some DNA-DNA proximity ligations to optimize secondary enrichment of chromatin-containing protein complexes. I want to give thanks to the Aitchison Lab, Sanjeev Kumar, Fred Mast, and then Peter Friddy and Mike Rout's lab who provided the nanobodies. Thank you.
Great. Thank you, Paul. And I'll just ask a quick question while everyone is unmuting themselves. I'm quite curious about your thoughts on how to overcome the technical challenge of distinguishing and isolating DNA. CTCF is just bound to DNA because it's bound to the DNA versus pulling down truly only CTCF that is associated with long-range interactions.
No, that's a great question. And what we need to know is in our grindate that we preserved these complexes by glutaraldehyde fixation. And we want to make sure that when we extract, we're also keeping these long-range interactions intact. And yeah, that's Yeah, that keep these long-range interactions intact, and yeah, I hope that we see some long-range interaction. I think I understood your question.
Yeah, well, is there a part of your method in which you are able to distinguish between simply CTCF bound to chromatin on the linear DNA versus the subfraction of CTCF that is associated with bringing long-range interactions together?
Well, we don't. What I've just shown you is just it's definitely just a short range. We haven't gotten to that point yet where we're going to be able to differentiate between the two.
Okay.
Yeah.
Anyone else?
Yes, this is Andy Belmont. I'd like to jump in on a similar question. I mean, I was intrigued by your microscopy, so I'm wondering if you've actually measured the size of the type of pieces you get. That's the first question. The second question is, we tried something years ago where we were kind of screwed because we were pulling down something with lac operators with GFP antibodies, GFP against repressor, but we found in our salt conditions we were getting large aggregates. So we were somehow getting aggregations of things that didn't contain lac repressor with other pieces that did. So you mentioned the large aggregates. So I'm wondering what the size of the small pieces are and how you try to minimize those small pieces coming together to form aggregates.
Yeah. So in that particular patient, we don't have the scale bar in there, but we've done other cryo grindings, and the pieces are sub-micron in size. To address the other question, yeah, I don't know how we're going to. Yeah, so we use this. So when we add the grindate to lysis buffer, we do a mild sonication to break apart all those particulates. And then we do centrifugation to clarify all that.
The last thing was that your nanobodies are going to be really useful for lots of applications. So will they be available? We've been using nanobodies for electron microscopy, and they are such a big improvement over normal antibodies.
Yeah, absolutely. So right now we don't have the CTCF, but we have the GFP, and if you contact me or John, we can get you the GFP ones, and then there's some other ones that may be of interest to you.
Okay, that would be great.
Hi, Paul. This is Bing Ren. I have a quick question. How many cells did you start in this experiment?
So I'll have to get back to you on the number of cells, but we use about 1 milligram of grindate, and really, but how many cells is that? That is the ultimate question. I use, so I do about 8 500 centimeter square plates of cells that are roughly 80% confluent. I harvest those. They go down into, I make the cell slurry. I get 5 mils of BBs out of that. And then we grind that, and we get about 5 mils of powder and 1 scoop. I'll have to do the back calculations to get back to you, but feel free to email me and I can tell you exactly.
Thank you.
But you have to start with a lot of cells to do the proper grinding. You can't just start with a 10 centimeter or a couple of 15 centimeter plates. You need lots of cells to do the grinding. Otherwise, you'll lose all your material. Just stick to the beads, and it doesn't grind properly.
Okay, great. Well, for the sake of time, I think we will